Welcome back to this next video, and in this video we are going to talk about the enzyme-linked immunosorbent assay, which is known as the ELISA. So the first thing is, what is ELISA? The enzyme-linked immunosorbent assay, the ELISA, is also known as the enzyme immunoassay or EIA. This ELISA is a plate-based assay technique which is designed for detecting and quantifying substances such as the peptides, the proteins, the antibodies, the hormones, any environmental sample. So you can actually detect a variety of things with the help of this ELISA technique. Now this ELISA, it was introduced in 1971 by Peter and Eva at the Stockholm University in Sweden. Now what happens in ELISA is that you are using an enzyme conjugated with an antibody. In this particular case, you will be having an antibody, as you can see over here. That particular antibody will be attached to an enzyme, and that enzyme is actually it will be acting on a substrate, which is a colorless substrate. And when the enzyme acts on this colorless substrate, you will be getting a color product, and that color product is actually telling you about the detection and the quantification of the target in your sample. So the enzyme conjugated with an antibody, that is usually called a conjugate when you talk about the ELISA, or sometimes it is known as the antibody conjugate. So the antibody conjugate simply means the antibody that has been attached with an enzyme. So in ELISA, an antibody conjugate that will react with its colorless substrate to give you a color product. Now the substrates that are used in the ELISA for these enzymes, they are called is the chromogenic substrates, because they are giving you the colored product when they are acted upon by the enzyme. Now a variety of the enzymes that has been used in ELISA, and the important enzymes that are used in ELISA, that includes the alkaline phosphatase, the horseradish peroxidase, and the beta-galactosidase. If you talk about the specific substrates, so the ortho-phenyldiamine dihydrochloride, that is actually acting as a substrate for the peroxidase enzyme like the horseradish peroxidase, and the para-nitrophenyl phosphate, that is actually a substrate for the alkaline phosphatase. Now these substrates that are used by these enzymes, they go for the hydrolysis of these substrates, and when they are hydrolyzed by the enzyme, they actually give you the colored end product. Now what you do in ELISA is that light absorption of the colored product that has been formed by the action of the enzyme, that is actually measured, and that absorption is converted to numeric values then in comparison to the controls that we will see in a while. Now the intensity of the color, or the optical density, that is actually measured at the 450 nanometer. So the light absorption that you will be detecting over here, which will be then converted into the numeric values, that is actually seen at the 450 nanometer. Now the two important things when you talk about the ELISA, these are known as the direct and the indirect. So you should have a very firm understanding of the concept of the direct and indirect in ELISA. So when you use this term direct and indirect in ELISA, these two terms, they're actually used in two perspectives. They are used with respect to the attachment, and they are used with respect to the detection. So the attachment can be a direct attachment, it can be an indirect attachment. The detection can be a direct detection, or it can be an indirect detection. So when you talk about this direct and indirect with respect to the attachment, so you are actually referring to the attachment of the sample to the microtiter plate that you used in the ELISA plate. When you talk about the detection, so you can actually directly or indirectly detect your target in your sample. So this sample term, that is a very general kind of the term, so that can refer to any kind of the sample that you are interested in. So here I'll be only using this term antigen just to represent this sample. Although this antigen is a very specific term, but we'll be focusing on a specific concept over here, and that will be true for all of the samples. When we talk about the direct attachment, indirect attachment, what you do is that a buffered solution of the antigen to be tested, that is added to the well. And when you add that to the well, that particular antigen is going to directly interact with the surface of the microtiter plate. So if you can see over here, if this is the well of a microtiter plate, the buffered solution of the antigen that you are testing, that is directly interacting with the microtiter plate. Therefore, it is called is the direct attachment. When you talk about the indirect attachment, what you do is first you attach an antibody to the surface of the microtiter plate. After that, you will be adding the buffered solution of the antigen to be tested to this particular well. So as you can see over here, your antigen that is actually indirectly attaching to the surface of the microtiter plate through this antibody, therefore it is known as the indirect attachment. So this one is the direct attachment, it is directly interacting with the surface of the microtiter plate. This is indirect attachment, where your sample or your antigen that are indirectly interacting with the surface of the microtiter plate. When you talk about the detection, and when you using the term the direct and indirect with respect to detection, what that means is, like say for example, if this is your antigen that has been attached to the microtiter plate, a single enzyme conjugate that is used, and that gives you the detection of the antigen in your sample. So as you can see over here, the antibody or the antibody conjugate, because this antibody has been attached to an enzyme that is directly interacting with your antigen, therefore it is known as the direct detection. When you talk about the indirect detection, as you can see over here, this is your antigen. First, a primary antibody, we will be calling this one is the primary antibody because this is directly interacting with your antigen. So in case of the indirect detection, what happens is that the antigen that is first bind by the primary antibody, and then you will be adding a secondary antibody, which will be actually the antibody conjugate, because the enzyme that is attached to this secondary antibody. So in this particular case, you are actually detecting your antigen indirectly with the help of this primary antibody. Therefore it is known as the indirect detection. So if you can say focus on these things here, the detection antibody is directly interacting with your antigen. Here your detection antibody or your antibody conjugate that is indirectly detecting your antigen. Therefore we are using the term indirect detection over here and the term direct detection over here. Now, when you talk about the types of the ELISA with respect to the term direct and indirect, there can be 4 types of the ELISA. The first type, there will be direct attachment and direct detection. In the second case, there will be direct attachment, but the detection that will be indirect. In the third case, there will be indirect attachment, but there will be direct detection. In the fourth case, the attachment and the detection, both of them, they will be indirect. So you will be having an indirect attachment and indirect detection. So we will be focusing on all of these, how these phenomena actually works. When you talk about the first type, the direct attachment and the direct detection, you are, as you can see over here, the antigen that is directly interacting with the microtiter plate without the help of the antibody. So this is actually showing you the direct attachment over here. If you can see over here, we are using a single antibody conjugate. On one side, this antibody is interacting with the antigen. On the other side, it is attached to the enzyme. So as a single primary and a single antibody conjugate that is used in the detection, so this will be the direct detection. So this type of the ELISA that will be known as the direct attachment and direct detection ELISA. The second type that is known as the direct attachment and the indirect detection. As you can see over here, your antigen is directly interacting with the surface of the microtiter plate. So the attachment that is direct. as you are using two antibodies for the detection. Therefore the detection that will be indirect. The primary antibody will be interacting with your antigen, the secondary antibody that will be interacting with the primary antibody, thereby giving you the detection of this particular antigen indirectly. Therefore in this particular scenario, the ELISA is the direct attachment and the indirect detection ELISA. If you talk about the indirect attachment and the direct detection, this particular phenomena is actually this is a special type of the ELISA, which is known as the sandwich ELISA, and you will see in a while why are we actually calling it as the sandwich ELISA. Now, if you can see over here, you are having these antibodies that are attached to the surface of the microtiter plate, and these are your target protein. These blue rectangles that you can see over here, that are your target protein. So as you are attaching these target protein or your antigens indirectly to the surface of the microtiter plate, so this is indirect attachment. Now, if you talk about the detection over here, this is direct, because as you can see over here, the antibody conjugate, we are using a single kind of the antibody. That antibody is attached to the enzyme. Therefore, you are using the antibody conjugate directly over here. So the antibody or the antibody conjugate that is directly interacting with the target protein. Therefore, the detection that is known as the direct detection. Now, why we call it is the sandwich over here, as you can see over here, your target protein that has been sandwiched between this antibody, the attachment antibody, and the detection antibody. Therefore, we usually call this is the sandwich ELISA, because your target protein or your antigen that has been sandwiched between the two types of the antibodies. So when the enzyme that is attached over here, you will be adding the substrate, and that is going to give you the color product. We will come to that in a while. So this one, the indirect attachment and the direct detection, you can actually call this is the sandwich ELISA. Now, if you compare it to the direct ELISA, the sandwich ELISA is much powerful, and it has a very higher specificity. The reason is that as compared to the direct ELISA, the sandwich ELISA, which combining antibodies to two different epitopes of the target protein, therefore it is actually having a higher specificity. This particular antibody, which is actually the attachment antibody that will be interacting with one type of the epitope of your target protein, or one kind of the epitope of the antigen, and this detection antibody that will be interacting with another type of the epitope. So the combining antibodies, and they are actually interacting with two different epitopes of your target antigen, thereby it has higher specificity. And this sandwich ELISA, it is useful for application that required higher accuracy, especially when we talk about the diagnostics. So the common ELISA that is used for the diagnostics, that is actually a sandwich ELISA. This sandwich ELISA, it is actually, you can see, of two types. In one case, you will be having the indirect attachment and the direct detection, but in the other case, you will be having the indirect attachment and the indirect detection as well. As you can see over here, this one will be your capture antibody. That captured antibody is going to immobilize or interact with the antigen or your target protein. So the attachment of the antigen or the target protein, that is indirect. Therefore, we call that is the indirect attachment. And as you can see over here is the target protein or the antigen that has been sandwiched between this antibody and this antibody. So you can call this is the sandwich ELISA. The difference from the first type is that in the previous type you were using a single antibody for the detection of this antigen or target protein. In case of the indirect detection, what you'll be doing, you'll be using two kind of the antibodies. The primary antibody that will interact with the target protein, the enzyme conjugate that will interact with the primary antibody, and the enzyme that will be converting the colorless substrate into a color product. So the difference between this sandwich ELISA and the one we discussed in the previous slide will be that you are using two antibodies in the indirect attachment and direct detection sandwich ELISA, and you are using 3 antibodies when you talk about the indirect attachment and indirect detection sandwich ELISA. So this is the concept and the types of ELISA with respect to the direct or indirect attachment or the detection. Now, if I give you a summary of the steps, what you do in the ELISA is that the first thing you have to do is that you have to pre-coat it micro well plate. So you can actually be preparing the wells of the micro titer plate for the attachment of the target protein or the antigen. Now, in this particular case, I'm showing you the indirect attachment method. So what you do is the first step will be the attachment of the capture antibody. This one is the capture antibody that is going to attach to the surface of the microtiter plate. If you're talking about the direct detection, you will not be using this capture antibody. So the first thing is you will be adding your capture antibody over here in case of the indirect attachment. Then you will be adding your sample or your standards. So when you add your sample, that particular sample is this antibody that will be specific. So the in the sample, your target protein that is going to interact with the capture antibody, and your antigen or your target protein that will be immobilized in the well. Now, one thing to keep in mind is that when you talk about this, the microtiter plate or the well of the microtiter plate, it can actually interact with the other proteins, especially the enzyme conjugate or the detection antibody. So when you add your sample, what you do next is that you are going for the washing of the well. And when you are washing the well, that means that any non-specific binding things that will be removed from the well, and only the target protein that will be in interaction with your antibody that will remain in the well, and the rest of these proteins or rest of these proteins in your sample that will be eluted from the well. So one of the important thing in the ELISA is that after each and every step you have to go for washing to make your results specific. So once you have added the antibody, the capture antibody over here, and then you have added your sample, when you wash the sample, when you wash this particular well, all of the unwanted proteins that will be removed from the well. Another important thing is that when you talk about the surface of the microtiter plate, it can bind with the detection antibody without specifically attaching to the antigen. So you have to ensure that the attachment of the detection antibody that is only to the target protein and not on the other surfaces of the microtiter plate. So what you do is that you will be blocking the attachment site, the other attachment sites on the plate by using other kind of the proteins. So in this particular case, say for example, this one is your capture antibody, and this is actually interacting with the target protein in your sample. And just for the sake of argument, there is another site also available over here to which the detection antibody can bind and give you false results. So what you do is that after the addition of the capture antibody and your sample followed by washing, you add the bovine serum albumin protein to the microtiter plate. What the bovine serum albumin do, that is going to block all of the attachment site on the microtiter plate, and the detection antibody will not be able to give you false results. So once you have added the capture antibody and the target protein that has been immobilized, and you have blocked all of other sites in the microtiter plate, the next step is that you will be adding the detection antibody. And this detection concept that will be true. You can directly detect your target protein, or you can indirectly detect your target protein. But in this particular image, you are seeing the indirect attachment. So what you will be doing is that you will be adding a primary antibody followed by washing, so that all of the primary antibodies that has not binded to the target protein that gets removed from the well. So when you have washed only the primary antibody that is attached to the target protein that will remain in the well, and all of the other primary antibodies that will be removed, because if they are there, the secondary antibody and the enzyme conjugated antibody that can bind to the primary antibody and can give you false result. But if you wash it, only that particular primary antibody that is in interaction with the target protein that will remain in the well. Then you'll be adding the secondary antibody, and when you add the secondary antibody, that is going to bind to the primary antibody, and after the addition of the secondary antibody, again you have to go for the washing so that to remove all of the unbound secondary antibody. And when you have washed that, after that you have to add your substrate to this particular well, and when you add the substrate to this particular well, the enzyme is going to act on that particular substrate, and that is going to give you a color product which you can actually absorb at 450 nanometer. So this one is very important, that you have to block all of the side of the polystyrene after the addition of your samples. This is actually a microtiter plate, and all of these yellow wells that are actually showing you the positive results. So when you are getting a yellow color in the well, that means you are getting a positive result, and in the wells you are not getting any kind of the color, that is actually showing you the negative results. If you like this particular video, please subscribe to my channel, hit the like button and share it with your friends. And in the next video, I'll be focusing on the how you will be differentiating between the positive and negative result in ELISA, and I'll be especially focusing on the concept of the competitive ELISA.